structures of DNA, RNA, mRNA and tRNA. So we want to look at, for the DNA, what are the components of DNA, how are these components arranged within the DNA double helix, and what is the function of DNA. So, first of all, looking at a nucleotide structure. So each DNA strand is made up of a lot of subunits, and each subunit is called a nucleotide. So this here is a mononucleotide. It's just one nucleotide. And in DNA, there are three components of a nucleotide. There is a phosphate group, a pentose sugar, and in this case, it would be deoxyribose. And you've got the nitrogenous base, A, T, C, or G. So adenine, thymine, cytosine, or guanine. And remember, A pairs with T, apples on the tree, and C pairs with G, cars in the garage. So key things here is there is a phosphate group, a pentose sugar, and a nitrogenous base. In deoxyribonucleic acid, the pentose sugar is deoxyribose, and the bases in deoxyribonucleic acid or DNA is A, T, C, or G. So you've got this mononucleotide. What can then happen is lots of these nucleotides can join together to form a polynucleotide strand, and they join together by a process called condensation. So deoxyribonucleic acid or DNA and ribonucleic acid, which is RNA, are important information carrying molecules. So in all living cells, DNA holds genetic information, and then the RNA transfers genetic information from the DNA to the ribosomes, the mRNA. And ribosomes are actually formed from RNA and from proteins. That's how they're made. And their function of ribosomes is obviously protein synthesis. And both DNA and RNA are polymers of nucleotides. So each nucleotide is formed from a pentose, a nitrogen-containing organic base, and a phosphate group. So they all contain those three components: phosphate group, a pentose sugar, and nitrogenous base. So looking in further detail at DNA now, DNA is a double helix structure, and it has two polynucleotide strands which wound around each other to form a double helix. And when you've got your mononucleotide, which would just be this here, these mononucleotides can join together to form a polynucleotide by a condensation reaction which forms something called a phosphodiester bond. So this would be a polynucleotide in which a few of the nucleotides are joined together by a condensation reaction, and because it's got ester bonds repeatedly, it's a phosphodiester bond. And the phosphate groups face outwards and they form a sugar phosphate backbone. So you'd have one polynucleotide strand going in one direction, and then another polynucleotide strand will form in the other direction by something called complementary base pairing. And in complementary base pairing, adenine binds with thymine and cytosine binds with guanine, and therefore you get two anti-parallel polynucleotide strands which will then coil up into a helix, and that is when you get your full DNA structure. So DNA is made of two polynucleotide strands joined together by hydrogen bonds between complementary bases, and the DNA forms like a ladder between the phosphates, and the deoxyribose and the phosphates form the sides. So you've got the sides of the ladder are your deoxyribose sugar and your phosphate, and then facing inward like the rungs in the ladder, you've got the nitrogenous bases by complementary base pairing, joined together by a hydrogen bond. So, the ladder-like arrangement of the polynucleotide chains is then twisted into a helix and wound around each other to form this double helical structure. And they form the structural backbone of the DNA molecule. And every complete turn of the helix, there are 10 base pairs. So we now just want to look in a bit more detail at the pairing of bases. So the organic bases contain nitrogen. So you've got your single ring shorter cytosine and thymine, and you've got your double ring longer adenine and guanine. And for them to be the same length, they must pair in a certain orientation. So apples on the tree, A binds with T, and then cars in the garage, C binds with G. So adenine binds with thymine, cytosine binds with guanine by complementary base pairing. And between the bases, there are hydrogen bonds. So there's 2 hydrogen bonds between A and T, and there's 3 hydrogen bonds between G and C. So if we now look at RNA, so RNA is single-stranded and it contains uracil instead of thymine. So the bases in RNA, instead of being AT, C and G, they're AU, C and G. So there's no thymine in RNA, there's uracil instead. So instead of thymine pairing with adenine, instead uracil pairs with adenine in RNA. And there's 2 types of RNA that you need to know about. There's mRNA, which is messenger RNA, and tRNA, which is transfer RNA. So messenger mRNA, or mRNA, mRNA is made during transcription, which happens in the nucleus if it's a eukaryote, or if it's prokaryote in the cytoplasm. So mRNA is made in transcription, and it carries a genetic code from the DNA, if it was a eukaryote, to the ribosomes, where it's then used to make a protein during translation. So it's a messenger because it takes the information from the nucleus to the ribosomes. And mRNA is a single polynucleotide strand. So in mRNA, groups of 3 bases are usually called codons. So transfer RNA or tRNA. tRNA is involved in translation. It carries the amino acids that are used to make proteins to the ribosomes. So it carries amino acids to the ribosomes in translation. And tRNA is a single polynucleotide strand. However, it gets folded into a 3-leaf clover type shape, or 3 hairpin loops. And that is because there's hydrogen bonds between specific base pairs which hold the molecule shape together in this clover-like shape. And every tRNA molecule has a specific sequence of 3 bases called an anticodon. So it's got these 3 bases which is called an anticodon, which it would bind to a complementary codon on the mRNA. So you've got these 3 bases of anticodon, and then you've also got an amino acid binding site at the other side for it to carry the amino acid. So we want to look now at the similarities and differences between the 3 types. So DNA, because it's got the double helical structure which protects the bases, it's a very stable structure. It's found in the nucleus, but it's also small amounts are found in mitochondria. A shape is double helix. Its size is relatively long. It can be up to 250 million nucleotide pairs long. The bases in DNA are adenine, thymine, cytosine and guanine, so A, T, C and G. The pentose sugar is deoxyribose, and each nucleotide contains phosphate, pentose sugar which is deoxyribose, and a nitrogenous base, which can be A, T, C, or G. And it is double-stranded, so it's got 2 strands. mRNA, because it's single-stranded, it's quite unstable. And it's found either in the nucleus, the cytoplasm, or ribosomes, depending on where it is on its journey. It's single-stranded, it's relatively short, and its bases, it contains A, adenine, cytosine, and guanine are the same, but there's no thymine. Instead, you have uracil. So you've got adenine, cytosine, guanine, and uracil. So A, C, G, and U. And the pentose sugar is a ribose. And each nucleotide contains a phosphate, a pentose sugar, which is a ribose, a nitrogenous base, which is A, U, C, or G. And it is single-stranded, so it's only got one RNA strand. Last one is tRNA, or transfer RNA. It's relatively stable, and that's because of its folded structure into the clover-like structure, or 3 hairpin loops. RNA is found in the cytoplasm, and it's involved in protein synthesis, because transfer RNA brings or transfers amino acid to the ribosome. It's got a distinctive folded structure with 3 hairpin loops that form the shape of a 3-leafed clover. One of these hairpin loops contains a sequence called the anticodon. It is the shortest of the 3 molecules, and the nitrogenous bases are adenine, cytosine, guanine, and uracil, so A, C, G, and U. There's no thymine. Pentose sugar is ribose, and each nucleotide contains phosphate, pentose sugar, which is ribose, nitrogenous base, A, U, C, or G, and it has one or single-stranded, it's one polynucleotide strand.